If you scream, think tyrosine. It's the source of catecholamines. So today I want to tell you about adrenaline and noradrenaline, aka epinephrine and norepinephrine, where they come from, and why amino acids are even cooler than you thought. So this is just a basic overview of where we're going. So don't worry if things seem scary now. Hopefully it'll be less intimidating in a minute. But the basic idea is that we can start from tyrosine or from phenylalanine. So both of these are amino acids. But so tyrosine can be made from phenylalanine. And so actually, I think half of the phenylalanine that you eat is used to make tyrosine. And so phenylalanine we can't make it though. So it's considered dietarily essential. So we can't make it ourselves. We need to get it through our food. That doesn't mean essential doesn't mean we don't need the other ones. We just can make them from other things. So we can make tyrosine from phenylalanine. And then we can use that tyrosine to go through a series of pathways to make L-Dopa, then dopamine, then noradrenaline or norepinephrine, and adrenaline or epinephrine. And so we'll go into all of these different steps and where the different names come from. And speaking of those names, let's first clear up some confusion about noradrenaline versus norepinephrine. They're the same. Adrenaline versus epinephrine. They're the same. Noradrenaline versus adrenaline. They're different. So these have adrenaline and noradrenaline differ in adrenaline has this extra methyl group. So this CH3. So as to the noradrenaline and norepinephrine and adrenaline versus epinephrine, those adding the epinephrine versus adrenaline or whatever that thing, that's all the same. And they both, so they mean the same thing, even etymologically. They just come from different from languages and were named by different people. And so there's different accounts of the early discoverers. So William Bates in 1886 found something produced by the adrenal gland. Then in 1895, a Polish physiologist, Napoleon Cebulski, found it. And then in 1901, Japanese chemist Yoshimi Takimine purified it from the adrenal glands, and he named it adrenaline, which means Latin for ad or near, plus renal kidneys. And then he patented it, and then Europe went with that. So they went with adrenaline. So if you're in Europe, it's commonly called adrenaline, noradrenaline. But then another scientist, John Abel, had already prepared an adrenal extract, and he called it epinephrine, which is Greek for epi, so on top of, and then nephros and kidneys. And so apparently it turned out that didn't even contain active adrenaline, but the US went with it. And so in the US, it's often called norepinephrine and epinephrine. So if you get confused about that, I'm sorry, and hopefully this helps clear up the confusion by at least showing you where that confusion can come from. So as to what they do, they both can act as chemical messengers, so sending signals, and they can act as neurotransmitters or hormones. And so basically, a neurotransmitter is where a neuron, so a nervous system cell, so cells going from your brain and stuff and throughout your body to send messages through neurons, which are really, really long cells. And then they can connect to either other neurons or to target cells, other cells like muscle cells, to send a message for your muscles to move, for your blood vessels to dilate or constrict or that sort of thing. And they send these messages, they can send little chemical messages that are really short range. So right between the tip of the end of the neuron and the start of the surface of the other cell. And so on the surface of the other cell, they'll have a receptor. And then that signal can bind to the receptor and cause a chain reaction within the cell. Or cause something like the cell to let something else in or something. So it has some So it's passing along the signal. And so typically these, so like without actually going into the cell, they can cause this like receptor to change, and then that causes changes inside the cell. So how is that different from a hormone? Well, a hormone is basically, instead of being released by a neuron, it's released by a cell it's released by a gland. And so these glands release their products. Instead of releasing them like just to that cell that they're really close to, they're going to release them into the bloodstream so they can go all throughout the body. But then only the cells that have the receptors for them will be able to respond. And they have different receptors. So there's a bunch of different like adrenergic or receptors or whatever that can bind to these different hormones, and they can have different effects. They're typically, you think of adrenaline with like your fight or flight response, doing things like getting your heart beat faster, your liver to stop storing glucose as glycogen and instead break down that glycogen, so promoting glycogenolysis, so you get more blood sugar. Can cause like constriction or widening of your blood vessels, depending on like, so you can direct the blood to places so that your body can be better in that, better able to respond to whatever like scared you or whatever, and constricted in like your fingers and stuff so you don't like waste your energy on, or like your toes, which don't need it as much. But basically there's a lot of like different actions, and I'm not a neurologist and I'm not an endocrinologist, and so I'm not going to try to get into too much of that. I just wanted to give you an idea. And so even though we think of like, especially adrenaline as kind of like this like fight or flight or whatever, noradrenaline, so noradrenaline is actually the main neurotransmitter that's used by the sympathetic nerves in the cardiovascular system. So it's really important. So you might have heard of like the parasympathetic as like the rest and digest, and sympathetic as like fight or flight. But you really need the sympathetic all the time, because like if your body was always getting the signal like, oh, relax, 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 you would like, your body would relax to the point of death. And so you really need the sympathetic nervous system in order to kind of keep things functioning. The adrenaline is made almost exclusively in this adrenal medulla, and it's getting signals to make it from like corticosteroids produced by the adrenal cortex. So these are, the adrenal gland is this gland that's on top of your kidneys. And remember, that's like the adrenal on top of kidneys, or epinephros on top of kidneys. And so it's going to make this molecule. And so whereas the, so like noradrenaline, well, so you can also get some noradrenaline from the adrenal gland, but you also get noradrenaline in your brain, but very little adrenaline, just in like a specific part of your brain. And you can also, another part of your brain will stop at the dopamine step and then be involved in like reward responses. And so these kind of different functions depending on like where they're being released, what cells are around, what receptors those cells have. All of these different things. But the reason why you're only going to get adrenaline, almost only going to get adrenaline in the cortex, is because if we look back at this pathway, the step to actually make the adrenaline from noradrenaline, it requires this PNMT enzyme. So I'll talk more about this later, but it's like this protein that helps speed up this reaction. And so the reaction, remember, we're transferring this, we're transferring a methyl group. We're actually going to transfer it from S-adenosyl methionine. So this is actually coming from another protein letter, methionine, we'll talk about. And so you're adding this methyl group here. And so only cells that express this enzyme are able to do that transfer. And so this enzyme is almost only made in the adrenal medulla. And so this is why you're almost only going to get it made here. There's also part of your small group of neurons in your brainstem that can make it. So you get some adrenaline made in your brain, but mostly in your nervous system, you're making the noradrenaline. And as I mentioned before, so this is regulated by the adrenal cortex. And so the adrenal cortex is also making a bunch of hormones, and some of these like cortisol, which you might have heard of as like a stress hormone. So it makes sense that your adrenaline would be controlled in part by messages from like things like cortisol. Okay, so those are that's like as far as I'm going to go into the biological like functioning of these molecules. And I want to tell you more about like how we actually make them and like the biochemistry of it all. Okay, so let's go back. And so we typically think about amino acids in the context of being protein letters. And so we have these 20 common amino acids, and they have this genetic backbone and this unique side chain that sticks out with these different properties. We can link these together, forming peptide bonds, as I talked about yesterday. You get these long chains of amino acids called polypeptides, and then those can fold up into a pretty protein structure. And the reason why they fold up into that pretty protein structure is because they have different sequences of R groups, so different unique side chains, and those side chains have different properties. And so they're sticking off and they're going to influence how the protein folds, because the protein's going to want to optimize the position of those. So like the negative ones will be by the positive ones, the ones that that really like water will hang out with the water, the ones that water doesn't like will get excluded from the water in this thing called the hydrophobic effect. But basically they'll get excluded by the water in this thing called the hydrophobic effect, where the water is going to kind of gang up around them. And that's going to influence the, but basically just know that they have these different properties. And so typically we think about those properties as helping determine their structure, but these properties also give them unique functions when to function outside of their like protein roles. And so this is how like we can get these peptide derived hormones. So there are different types of hormones, and so some of them are made from peptides, and or like we call them like peptide or amino acid derived. And so these include the catecholamines that we're talking about today. And so these are made from, specifically, they're made from tyrosine, which remember can be made from phenylalanine. So this ring thing, this is what we call, so this is a benzyl. So you have this resonance stabilized ring. Okay, what do I mean by that? So atoms link up by form bonds by sharing pairs of electrons, and much more on this in previous posts. But so typically you think about like a single bond being sharing one pair, a double bond would be sharing two pairs, and then electrons can also exist as lone pairs where they're not being shared. Sometimes they're shared communally, so more than two atoms are sharing, and so you get like partial double bonded character like everywhere. And so one place this occurs is in these aromatic rings. So you have these, the electron density is like delocalized around these rings. And so looking at the difference between phenylalanine and tyrosine, the difference is just this OH group. So you can go between these with this phenylalanine hydroxylase enzyme. And so hydroxylase, so it's adding an OH group from molecular oxygen. And so this OH group is actually makes it much better for being a hormone, on the way to being a hormone at least, because now it is more soluble because it has this OH group. So if you look at phenylalanine, so in order for something to be soluble, it has to to be soluble means it has like each like copy of it has its own full water coat, or a full coat of the solvents. And so typically we're talking about water here as our solvent. So the thing that's dissolving something, and then the thing that's dissolved is your solute. So here we're talking about like tyrosine being our solute and water being the solvent. So we call water-based solutions aqueous, and they're typically what we're dealing with in like biochemistry and stuff. Although like if you had something that was like a membrane protein or whatever, you could talk about it being lipid soluble. But we're going to be, when I say soluble, I'm talking about water soluble, and that will allow it to like travel through the bloodstream and stuff. And so if you have something that's hydrophobic, so that water doesn't want to hang out with, it's going to be the water is not going to want to give up its water-water interactions to hang out with it. And so basically the hydrophobic thing is not going to be soluble because the water doesn't want to make bonds to it. And so instead, these will kind of clump up together, and then the water will cinch around it to minimize its contact. So we have phenylalanine is very nonpolar, it's very hydrophobic. So by nonpolar, that means like the electron density is like shared pretty evenly. So when we talk about atoms sharing pairs of electrons to form bonds, if they don't share fairly, then you get regions that are partially positive and regions that are partially negative, and then we call this polarity, and we call this a polar molecule. If all of the atoms are sharing fairly, then you don't have an imbalance of charge anywhere, and so you have a non-polar molecule. Water is really polar, so it's going to like other polar things because it can like other water molecules, because it can put its partly negative parts by its partly positive parts, and form these like intense water networks, or extensive and not intense water networks, and that can allow for these like water's really happy with itself, and it can dissolve a lot of things because if those things are polar or they have like full charges, so we call fully charged things ionic. But so if we look at our phenylalanine, we can see that its side chain, it just has like carbon and hydrogen share pretty fairly. And so this is going to be very nonpolar and hydrophobic. If you add this OH group, so oxygen is really what we call electronegative. So it's going to hog electrons. So it's going to hog the electrons that it's sharing with the other atoms. And this is going to make, so now you have this oxygen be partly negative and like this hydrogen be partly positive. And now you have something that water is okay with. So it's still not super water soluble, but it's better than phenylalanine. And to make it even better to travel through the bloodstream, to make it even more water soluble, we can add a second OH group. And so this is done by this tyrosine hydroxylase enzyme. So remember hydroxylase, we're adding this hydroxyl, so this OH group. And this is done with this tyrosine hydroxylase enzyme. And it's going to add this second OH group. And when you have a benzene ring with these two OH groups, we call it a catechol. So that's where that cool name comes from. It's because it has this benzene with these two OH groups. And at this point, when we call these catecholamines because remember we have this amine group from our amino acid. So if we go to our functional groups, we have an amine group, and we have this catechol group, which isn't on this chart, but we can see that it's a benzene ring and it has these hydroxyl, this OH group, two OH groups. So that's why we call these catecholamines. And you can see that although we're going to make changes, we're not going to change like this bottom part. So we're not going to take this away from being a catecholamine, and we're not going to change. We're not going to take away this nitrogen. So we can call all of these catecholamines. And so we're done this. So now we've encountered two of these two different enzymes already. Although if you start with tyrosine, then you don't need this one. But these both are using tetrahydrobiopterin as a cofactor. So a cofactor is basically something that an enzyme uses as a helper. So an enzyme is a reaction speeder upper. It can do things like hold the reacting molecules in the optimal position to react and stabilize uncomfortable transition states, provide give and take electrons, hold on to things, all sorts of different things. And so sometimes in order to do their work, they use, they bind to other molecules to help them out. And we call these other molecules cofactors. And I really need to organize my figures ahead of time better. So we call those cofactors. And so we can refer to, if an enzyme has a cofactor, we can call it, when it's alone, we call it an apoenzyme, enzyme by itself. But then when it has a cofactor, now it's a holoenzyme, so it's all whole. And so sometimes these are metals, sometimes these are organic, which are often vitamin based, as we'll see in a couple in this case. And so these are cofactors can provide additional opportunities, so additional functional groups and different metal groups to help bind things better, all sorts of different things. So enzymes often use cofactors is the basic point I wanted to make. And so in this case, they're using this cofactor called tetrahydrobiopterin. Okay, so we've gone from our tyrosine now to L-Dopa. Okay, so L-Dopa, so the L, so this goes back to our discussion of chirality, so handedness, so the right hand versus the left hand, non-superimposable mirror images. And so almost all of the amino acids you'll encounter in biochemistry, except for some cases with some bacterial wall stuff, are in the L form. And so that's where this L comes from. So you have it attached in a way. You have this group, so you can see that this carbon, this alpha carbon, that's attached to 4 different things. If you have a carbon attached to 4 different things, then you have a chiral center. And so you can think of this as being stuck, so it's sticking out at you, or so it's sticking back. And one version would be L, one version would D, and all of the amino acids that we use are L. And so that's where the L part comes from. Okay, so now we've made a catecholamine. We have this L-Dopa, but we now want to make it a neurotransmitter or a hormone. And so we've made it more soluble, but now we need to do more stuff to it. So the next step is done by another enzyme, dopa decarboxylase. And so carboxyl, so remember, carboxyl group is going to be a C double bond to an O attached to an oxygen. And then this could be protonated or deprotonated. So we call those different forms, carboxylic acid or carboxylate, depending on whether they're protonated or not, but it's the same molecule that can be just, yeah. And so if we have a decarboxylase, it's going to remove a carboxyl group. So it's going to remove this carboxyl group. So we're going to remove the carboxyl group of the amino acid. So now it's no longer going to be an amino acid, but it still has that amino group, so it can still be a catecholamine. And so this decarboxylase is going to use a cofactor too. And it's going to use a cofactor called pyridoxal phosphate. You'll see this PLP a lot of places in biochemistry. And so if you've been following me a while, you might remember in the talk about the amino acid lysine. So lysine is another amino acid we've talked about. And so it's this. And it can form what we call shift bases. So it has this amino group at its end, and it can form these shift bases. So basically it double bonds to a carbon. And this allows it to kind of hold on to this cofactor PLP, which is the cofactor we're talking about here. And so in this case, we were talking about lysine being used in transamination. So like moving an amine group from one place to another, which is involved in helping make amino acids from like sugar breakdown products and stuff. But basically, this shift base allows it to hold on to this PLP. And PLP doesn't just have to do this transamination. It can do all sorts of other things. And so one of the other things that it does is helps out with this decarboxylation reaction. So yeah, I just got this from this. There was this review article about all the different things that PLP could do. So you can see that it's forming this shift base. So it's holding on to that PLP like we saw before, but then it's going to switch that out so the enzyme is going to kind of hold that PLP in place. And then once the substrate comes in, so the L-Dopa, now it's going to hold on to that and promote the release of this carboxyl group as CO2, then let go. And so now you get dopamine and you've regenerated your enzyme. So another thing, so pyridoxal phosphate is actually derived from vitamin B6. So remember I said that sometimes the cofactors were often derived from vitamins. And so when we talk about vitamins, typically like B6 is actually means like there's like a whole family of different compounds that are similar but not identical that can all be considered B6, and some of them can be like interconverted between them and stuff, and it gets really confusing. But so basically this is a product of B6, and you also this dopa decarboxylase, so this DDC, it also decarboxylates other aromatic amino acids. So remember, aromatic means it has these rings. And because of this, another name for it is aromatic amino acid decarboxylase, or AADC. Okay, but the result is you get dopamine. And so some cells in their brain, so in a part called the substantia nigra, will stop there. They'll use dopamine as a neurotransmitter. And this is involved with like reward responses. But most of the cells will take things further. And the next step is going to be to do another hydroxylate, add another hydroxyl group. And so they're going to do it here. And this is going to be done with the help of dopamine beta hydroxylase. And so it's going to use ascorbic acid, which is vitamin C. And so it's going to hydroxylate here. And this is going to give you noradrenaline and norepinephrine. And so remember, this is going to be used extensively as a neurotransmitter and some as a hormone. And most cells will stop here, but not in your adrenal gland, in your adrenal medulla, where they have the enzyme that they need in order to do the conversion. And so that enzyme that they need is this phenylethanolamine and methyltransferase, or PNMT. And it's going to use a cofactor too. And it's going to use a cofactor that I've talked about before too, S-adenosylmethionine. And so as the name suggests, this is coming from methionine. And this is it's coming from the amino acid methionine. So now we've talked about phenylalanine, tyrosine, lysine, and now we're on methionine. So methionine, it has the sulfur, and you can see that it has this methyl group at the end after that sulfur. And it's kind of like begging to be taken off, isn't it? And so a methyltransferase is able to actually, methionine adenosyltransferase, or MAT, is able to take methionine, take ATP, and actually put this methionine onto this this adenosine part. So not the phosphates, but the phosphates will get released, and so this is going to provide energy to drive the reaction. And so this now is attached to this. And now you have a charge here, so you have something that's going to be more reactive. And a methyltransferase is going to help now transfer this carbon group to something else. And so now you can see that the sulfur, now you have lost the charge. So this is happier. And now your thing is methylated. And then the whole thing can be like regenerated. And so in this case, what we're methylating is we're methylating this amine group. And so this now we have adrenaline, and this can be made by in your adrenal medulla and a few of those cells in your brainstem, and be really important for sending messages. And it's also important that you're able to degrade those messages. And so breaking down the catecholamines requires this, like catecholamine O methyltransferase, or COMT, and monoamine oxidase, or MAO. And so you might have, MAO might sound familiar, and it's likely because MAO inhibitors can be used in some antidepressants. One final note, tyrosine can also be used to make thyroid hormones. And this is one of the only places in biochemistry where you'll see a kind of weirdo element. I don't know. But you don't see iodine very much in biochemistry, but you do see it in your thyroid hormones. So tyrosine can be like iodide, whatever, have iodine groups stuck onto it. And so, yeah, so tyrosine can be used to make a lot of different things. And other amino acids can be used to make other hormones. So like tryptophan can be used to make like serotonin. So there's all different uses of amino acids outside of just being for protein. So we've seen how we can have them being made to make hormones and to make like cofactors, like how methionine is used to make this SAM. And so, yeah, so amino acids are even cooler than you thought.